The 2019 novel human coronavirus, which has later been renamed as SARS-CoV-2, is the causative agent of the ongoing pandemic of coronavirus disease 2019, known as COVID-19, manifesting severe lower respiratory tract infection and acute respiratory distress syndrome. What is the difference between epidemic and pandemic? Epidemic is a rapid outbreak of an infectious disease in a given population that restricts to a particular location, also within a short period of time. But pandemic is a different thing. It's basically an epidemic situation that occurs worldwide over a very large area, crossing the international boundaries, usually also affects a large number of people. The novel coronavirus originated from Wuhan in China in late December 2019, and eventually it spread across the entire globe.
So, what are the clinical features of this COVID-19? Patients can be either symptomatic or asymptomatic. The symptomatic ones suffer from cough, fever, diarrhea, and myalgia. In worst cases, a patient might end up suffering from septic shock, renal failure, pneumonia, which might finally lead to death. While the asymptomatic people don't show any of these symptoms, but they are capable of transmitting the infection.
Now we will discuss about some interesting features of the SARS-CoV-2. As you can see the image, the SARS-CoV-2 belongs to the Coronaviridae family and Coronaviridae subfamily. They are under the genus of beta coronavirus. We all know that viral genome is DNA or RNA in nature. In case of coronaviruses or SARS-CoV-2, the genome is single-stranded RNA, which is 30 kilobase pair in length. The SARS-CoV-2 has also some structural and non-structural proteins. The structural proteins include nucleocapsid, spike, envelope, and membrane proteins. And the non-structural proteins range from non-structural protein 1 to non-structural protein 16. So, why the name is corona? As you can see in the electron microscopy photograph, the structural protein gives it a town-like appearance. That's why it is named as coronavirus. These coronaviruses are not new to the human beings. There are commonly circulating coronaviruses like 229E, HKU1, NL60T that creates the seasonal or cyclical infection. Eventually, these commonly circulating coronaviruses were mutated to severe acute respiratory syndrome, which appeared in 2002 in China, and later it also appeared in 2012 in Saudi Arabia in the form of Middle East respiratory syndrome. Later in late December 2019, it appeared as the SARS-CoV-2. Now, we will discuss about the transmission cycle of this SARS-CoV-2. It has been found that the genome sequence of SARS-CoV-2 is almost 96% identical to rhinolophus bats, indicating that bats are the potential reservoir of this SARS-CoV-2. From these bats, some small mammals like pangolin did harbor the viruses and plays the role of intermediate host. Incidentally, some humans got these viruses from pangolin, and from these humans, droplet or via some other direct contacts mode, other humans got infected and human to human transmission started.
Now, my colleague will explain to you the SARS-CoV-2 structure.
So this is the SARS-CoV-2 model. Viruses, they are generally made up of two types of proteins, structural proteins and non-structural proteins. The structural proteins here are red one, the spike protein, yellow one, the envelope protein, and green one, the M proteins. Inside also, the red ones that you are seeing are the nucleocapsid proteins. The virus enters the body through the eyes, nose, and the mouth. It searches for the ACE2 receptor on the cell. As soon as it finds the ACE2 receptor, it gets attached to this ACE2 receptor and moves into the cell. When it moves into the cell, it releases its genetic material, which is the RNA. Here comes the role of non-structural proteins. Non-structural proteins, they again form more such kind of RNA molecules, and along with the structural proteins, which form the outer part of the virus, more number of viruses are found. And then these are released from the cell. As you can see here, there are more number of viruses. These viruses are now capable to infect the cells which are beside the infected cell. And this is the way the virus spreads throughout the body. Thank you.
Now that we know how the virus replicates, let's talk about how the virus spreads in the environment. Now, when a person coughs or sneezes, large number of droplets are spread into the environment. These droplets are of varying sizes, with the smallest droplet nuclei spreading up to 150 meters far away. Apart from this aerosol transmission, these droplets can infect a susceptible person either directly by contact or indirectly through sharing of common items. Extensive research has been carried out to find out how long coronaviruses can remain on different types of surfaces. It was seen that the virus lasts only a few hours on objects like paper, while it survives the longest on the outside surface of a mask. So how do we combat this disease? There are ongoing treatment regimens being followed, including the prescription of antiviral drugs such as remdesivir, favipiravir, ivermectin, among others. And there are also numerous vaccines that are in the making against the virus. And the subject of vaccines will be talked about largely by my colleague later on. While the drugs and vaccines are still being tried and tested, there are certain things that we can do ourselves at an individual level, simply by maintaining basic hygiene. One of the most basic ways of maintaining proper hygiene is washing our hands, that too with soap. Now, what's so great about soap? Well, soap works great on anything that's oily. Viruses, in fact, are oily too. They have a layer of lipoprotein that the soap acts on and effectively disrupts, as a result of which this virus gets inactivated. So that's why washing our hands with soap is so important. But washing our hands with soap is not the only way to take care of ourselves. There's another extremely important measure that we need to take, and that is wearing a mask. As you can see from this image, when both persons are wearing a mask, spreading the virus from an infected person to a healthy person becomes so easy as compared to when either of the person is wearing a mask. And the best way of prevention is seen when both the individuals, the infected and the susceptible individual, are both wearing masks. And this drastically reduces the spread of the virus. Now, we've all seen people wear masks in different styles. But you know what? There is a very effective way of wearing a mask that can prevent you from coronavirus disease. Well, obviously, we need to wash our hands first thoroughly with soap. Then, we need to inspect the mask for any kind of damage or any dirt particles that may be there. After which, properly you can adjust the mask to your face, making sure that there are no gaps present. The nose, mouth and chin need to be covered effectively. And definitely, you must avoid touching the mask once it's on your face. While removing the mask, you again must clean your hands and carefully remove the mask by the straps behind the ears. You should pull the mask away from your face and store it in a clean place. And never forget to wash your mask regularly. And definitely wash your hands after use. So now that we know what's the right way to wear a mask, there are certainly things that we should avoid while wearing it. Wearing a damaged or loose mask is a strict no, and the mask should definitely not be worn under your nose. Always keep the mask on in front of people, even at 1 meter distance and closer. And most importantly, although we say sharing is caring, you must never share your mask with others.
Let us now discuss about the COVID-19 testing facility at ILS. With due approval from the competent authorities, ILS officially started the COVID-19 testing from April 14, 2020 with just 44 samples. The dynamic team for COVID-19 includes 3 virologists, 4 molecular microbiologists, 3 molecular biologists, 25 research scholars, 15 laboratory technicians and support staff. During the course of our COVID-19 testings, we have been applauded by several organizations. We have the capacity to conduct approximately 1,000 tests per day. Till date, we have conducted 1.3 lakh tests from 26 districts of Orissa, contributing to nearly 15% of the RT-PCR tests. Till date, we have reported more than 7,200 positive cases from the state.
ILS has a state-of-art BSL-3, that is Biosafety Level 3 laboratory used to study infectious agents that can cause potentially lethal infection. At ILS, some of the procedures required for testing of COVID-19 patient samples are performed in the BSL-3 facility. The BSL-3 facility is equipped with biosafety cabinets that carefully control the airflow to prevent exposure of the personnel to the infectious pathogen. BSL-3 facility uses controlled or directional airflow to ensure that airflow from non-laboratory areas into laboratory areas as an additional safety measure. For this purpose, the lab is maintained at negative pressure, which ensures that nothing from the laboratory escapes to the outside environment. The exhaust from the BSL-3 lab is sterilized by passing through the microfilters that remove pathogens and other harmful agents. Other engineered safety features include the use of self-closing or interlocked doors, sealed windows and wall surfaces, and a filtered ventilation system. BSL-3 labs also have access to equipment like autoclave to sterilize and decontaminate the laboratory waste. We receive the COVID-19 swab samples in an enclosed tertiary container, which ensures there is no leakage of samples. These boxes maintain the cold chain to keep samples viable for longer time during transportation. Once sample boxes are received at ILS, are then surface decontaminated by a solution of bleach and Lysol. After surface decontamination, sample boxes are sent inside laboratory via UV pass box, where it is received by trained personnel completely equipped with personal protective equipment or PPE. These boxes are then opened inside biosafety cabinets and samples are taken out, surface desterilized and arranged according to identification code given by the sample collection centers. Once samples are arranged, processing team takes over and start processing the samples for extracting the RNA. All the above processes are done in biosafety cabinet with trained personnel wearing PPE and N95 masks. RNA isolation is done with automated RNA isolation system. The isolated RNA is passed through pass box to the PCR team to set up PCR reaction to detect SARS-CoV-2.
There are three main types of COVID-19 tests. The nucleic acid test, antigen test and serological test. The nucleic acid and the antigen tests detect the viral RNA and viral protein respectively, whereas the serological test is performed to detect antibodies that are developed in a patient when they have been infected with the SARS-CoV-2 virus. Here at ILS, we perform the RT-PCR test, that is the real-time polymerase chain reaction, where the samples from oral and nasal swab samples are subjected to RNA extraction followed by reverse transcription. This reverse transcribed RNA is then subjected to RT-PCR amplification, where an enzyme DNA polymerase will amplify the primer attached to the viral gene. This enzyme not only has the ability to amplify the primer, but also it detaches the probe from the quencher. This probe then sends a fluorescent signal to the RT-PCR machine. This machine will then detect the signal and show the amplification, as you can see below, in the positive sample, and if there are no amplification, the sample is considered to be negative. Here representative figure showing a infected and non-infected sample tested for SARS-CoV-2. The positive sample shows amplification for two genes, that is the N gene shown in green and ORF1 gene represented in blue. However, there is no amplification in the negative sample. This is a representative video on the setting up of the RT-PCR test. This is done in the BSL-2 facility. Here, we are adding the master mix into all the 96 wells of the reaction plate. Using this plate, we can analyze 94 patient samples and include one positive and one negative control. After receiving the RNA plate from the BSL-3 facility, using a multi-channel pipette, we add the RNA into the RT-PCR plate. The multi-channeling pipetting ensures that there is no sample-to-sample contamination and there is addition of equal volume of RNA in all the wells. Next, we seal the RT-PCR plate using an optically clear adhesive film. This optically clear adhesive sealing is important as it ensures three things. First, it ensures that the machine is able to detect the fluorescent signal from the plate. Second, it ensures that there is no well-to-well spillage. Third, it prevents the evaporation of the reaction mixture from the RT-PCR plate as the temperature in the instrument can go up to 95 degrees Celsius. The plate is then mixed properly using a vortex and spun in a centrifuge to bring all the reaction mixture to the bottom of the plate. This is a RT-PCR instrument which is attached to a software where we provide all the necessary patient information and PCR cycling conditions. The prepared PCR plate is now inserted into the RT-PCR machine, which will now record the data for subsequent analysis. Here is an example of how the machine detects the amplification of the viral genome in real time, justifying its name. You can see the amplification of one positive sample and one negative sample in this video. Here you can see how the result looks after the amplification is done for all the 94 samples.
What are vaccines? Vaccines are substances that partly resemble a pathogen, which when injected in our body train our immune system to detect and attack invading pathogens. Our immune system is a complex network of molecules and cells that have ability to detect and degrade pathogens. The part of the immune system that the vaccines train is the adaptive immune system, which comprises of T cells, B cells and antigen presenting cells. The adaptive immune system first recognizes an invading pathogen, and after detecting the pathogen, the antigen presenting cells present the antigen to T cells, which activate the B cells, which eventually mature into antibody producing plasma cells that rapidly divide and grow to expand and produce copious amounts of the antibody. The phagocyte cells, which are macrophages, engulf the antibody coated pathogens and degrade them, whereas the killer cells detect the infected cells via the antibodies and kill the cells. To prevent against future infections, the adaptive immune system develops memory T cells and memory B cells that rapidly respond to a secondary infection by the same pathogen. Overall, this is called the immunity, and the vaccines work safely by training the adaptive immune system to eradicate or to create immunity against specific pathogens.
These are some of different kinds of COVID vaccines in pipeline like BNT162b2 from Pfizer and mRNA-1273 from Moderna are using mRNA-based technologies and are under consideration to be used in Western countries. Covaxin by Bharat Biotech in collaboration with ICMR is attempting to use inactivated SARS-CoV-2 as vaccine. GSK is using virus-like particles to develop vaccine, and Novavax is using recombinant spike proteins as vaccine candidate. In addition to these, there are many other vaccine candidates based on different technologies undergoing clinical trials to determine their efficiency and safety in humans. COVID-19 related activities at IISF include development of antibodies against SARS-CoV-2 to aid in virus detection and enable development of diagnostic kits. We are isolating and propagating the SARS-CoV-2 clinical strains to facilitate COVID-19 research and drug discovery. We are sequencing the genome of circulating virus strains and studying immune responses and immune profile of COVID-19 patients to identify factors that promote disease severity or offer protection. We are attempting to repurpose approved drugs and identify plant products with potential to inhibit SARS-CoV-2. We are also working on developing animal models to enable disease pathogenesis and challenge studies. Cysteine as metagenome profiling in COVID-19 patients is being done to correlate macrome diversity with disease severity. We have established biorepository for COVID-19 patient samples to enable research on COVID-19. Significant highlights of COVID-19 research at IISF Bhubaneswar include sequencing of 225 viral genome and identification of 5 clades prevalent in Orissa and the dominance of clade 20A and 20B over time in Orissa. We have identified D614J mutation in spike gene of most of the circulating viral strains in Orissa. This mutation is reported to increase the viral fitness by promoting its infectivity and transmission. We have isolated and propagated 17 different virus strains from patient swab samples. Evidence has been found for the fecal dissemination of virus suggesting of other probable mode of transmission and infection.
खुद को बचाना है तो दूरी बनाना है, साफ रहेंगे हाथ तो सही रहेगा स्वास्थ्य। ये उपाय है सबसे खास, चेहरे पे लगाए सदा मास्क। कोरोना संक्रमण को जो है हराना तो भीड़ को बिल्कुल नहीं जुटाना। रहोगे सावधान तो पाओगे समाधान। जिंदगी से है प्यार तो मिलने जुलने से करो इनकार। जिंदगी है प्यारी तो कोरोना से लड़ने की करो तैयारी। हाथ रखो अपने साथ, सैनिटाइजर रखो सदा पास। मास्क है जरूरी और भीड़ से रखो दूरी। व्यायाम और पौष्टिक खाना अब हम सबको है अपनाना। विटामिन भी जरूर खाइए, प्रतिरोधक क्षमता अपनी बढ़ाइए।